Welcome back. We have last session. Towards the end, we have showed you the structure of thiamine pyrophosphate, which is the coenzyme form of vitamin B1, that is known as thiamine. Again, we can show the slide where we ended, that this is the thiamine, which has got a pyrimidine ring and the thiazolidonium ring, and it is present as the pyrophosphate in the coenzyme form, and we have discussed the various structural aspects, that it can exist in zwitterionic form, which is also stabilized by the 3D orbitals of the sulfur, and here you can also have the carbene form. Okay. So, these are the two forms, but this is the predominant form, and the very fact that this is the predominant form makes this carbon very nucleophilic. Okay. So, if there is an electrophilic carbon, then it can react with that electrophilic carbon. Now, the reaction that the thiamine pyrophosphate participates, that means it helps the enzyme to carry out reactions, many reactions, but one very important reaction which we are going to discuss is the formation of what is called acetyl coenzyme A, CH3 COS-CoA. Now, acetyl coenzyme this coenzyme A is the building block for biosynthesis of various fatty acids, long chain fatty acids, and then also the building blocks for making the steroids. So, it is very vital, steroids like cholesterol, and from cholesterol you get different types of hormones. Okay. So, formation of acetyl CoA is biologically very important. Now, what is acetyl CoA? That CH3 structure is given, C double bond O, and this is SCOA. That means, it is a thioester of what? Thioester of coenzyme A. This is the structure of coenzyme A, SH, or you can write it COASH. Okay. So, it ends up with a sulfur. So, it has got a sulfur sulfhydryl SH arm, and that is a very good nucleophile, that we know. So, it is a thioester of coenzyme E, and thioester of what? What is the acid component? Is the acetic acid. So, it is called acetyl coenzyme A. Now, what is the starting point of acetyl coenzyme A? Now, this is the retrosynthesis or retrobiosynthesis. If you use that, so this, what is the source of this acetyl coenzyme A? It has been found that the source is nothing but the product of the metabolism of glucose, that means the glycolytic cycle, where the glucose is broken down to the final product is the pyruvate. Okay. So, this pyruvate, by a decarboxylative oxidation, because if you decarboxylate this carbon, you have to decarboxylate it, because this is a 2 carbon unit and this is a 3 carbon unit. So, the carboxy has to be lost. So, it is a decarboxylation, and however, decarboxylation should lead it to the aldehyde stage, but it is not in the aldehyde stage. This carbon is in the acid stage. So, it has to further be further oxidized. So, it is a decarboxylative oxidative process. Okay. And this reaction needs actually various coenzymes, not only just thiamine pyrophosphate. It is a quite complicated reaction, although the mechanistically it is very simple organic chemistry, but there are 3 different coenzymes are involved in the process. First of all, first coenzyme that is required is what is called thiamine pyrophosphate. Okay, that is what we are discussing. So, thiamine pyrophosphate first does the decarboxylation, and then there is another coenzyme which is called lipoic acid. Lipoic acid, I have shown the structure earlier while discussing, I showed the structure of these different B complexes, different B complexes like, no, maybe it is here. I will show you the structure. Where is that? No, not here. I will just write it. Okay. So, there is another coenzyme which is called lipoic acid. The structure is shown here. This is what is lipoic acid. It is a C8 8 carbon fatty acid, but containing a disulfide bridge. It is a 5-membered disulfide bridge. That is what is lipoic acid. So, the second coenzyme that is required is what is the lipoic acid, and the third coenzyme is what is called flavin adenine dinucleotide. And the structure, all the structures are given here. This is lipoic acid and this is thymine pyrophosphate, and this is what is flavin. Flavin moiety is this one, flavin adenine, flavin is up to this point basically. Riboflavin, you know, riboflavin is the vitamin B2. So, that is converted into the active coenzyme form by the formation of a dinucleotide, because there is one type of base here, sugar and then phosphate, and there is this base. This is a sugar also, ribitol and the phosphate. So, flavin adenine dinucleotide, FAD. So, 3 coenzymes are needed to carry out this reaction, and I will show the mechanism slowly, step by step. Okay. This is what is the mechanism, but I will definitely I can show it later once I describe this. Okay. So, let us describe, writing is the best way to understand. So, you write the structure of this TPP, thymine pyrophosphate. We will simplify the writing of the structure, okay, but we will write this phosphate, and this is methyl, okay, and this is the minus and that is plus, and your substrate is pyruvate. Okay. Remember, your substrate is a pyruvate or pyruvic acid, and then the reaction that takes place, as I told you, that if there is a good electrophilic carbon, then this can act as a nucleophile and can form this. Okay. Because this alpha carbonyl moiety in the pyruvic acid is very electrophilic because of the presence of this electron withdrawing carboxylic acid. Okay. So, you have Rn, then double bond, then S, then this, and then OPP, and here what you are having now CH3, and this takes up a hydrogen. As it goes, it takes up a hydrogen, most likely from the enzyme active site amino acid, which is having a conjugate, which is having a basic site, and that is present in the conjugated acid form. Okay. And then that can be donated. So, what I am saying is that BH plus, the B is having the plus charge. So, that takes the hydrogen and the B is released as the base. Okay. So, that becomes OH, and then you have this C double bond O OH. Now, just like you have a scenario in pyridoxal phosphate where there is an electron sink, it can act as an electron sink, and after acting as an electron sink, it acts as an electron source. The same thing happens here, because now if this comes back, this decarboxylation, because of the presence of this electron withdrawing group at the beta carbon. So, this is the alpha carbon with respect to the carboxy, this is the beta carbon. So, you have this double bond N plus. So, it is a very good electron withdrawing group or electron sink. So, then this type of process can take place. So, what will happen now? This is the decarboxylation. So, you get minus CO2, the nitrogen becomes neutral, and this is the substitute. These are the substituents of thymine pyrophosphate, and here you have double bond C methyl and then OH. Now, again what happens here, that as nitrogen is acting as the pulls up the electrons, this whole system loses the aromaticity which it had because of the sulfur lone pair participation in this ring, completing the 6 electron Huckel system. Okay. So, it again wants to regain the aromaticity. So, it goes back, and then now what happens? So, making this carbon quite nucleophilic. So, now there could be, if there is another electrophile, now this electrophile is basically in the form of lipoic acid. 1, 2, 3, 4, 5, 6, 7. So, one more CO2H in the form of lipoic acid. So, this attacks this sulfur, and the disulfide bond is very vulnerable. So, in disulfide, even if you add say a hydride reagent, so you get the SH, means the hydride attacks this sulfur and this goes and takes the hydrogen. So, this acts as a nucleophile and attacks this sulfur, and then this one goes out. Okay. So, the product is now N double bond S, this is the structure, this is R. Now, this becomes again plus, and then this is methyl OH, and then you have this S, and then I can just write this is SH, and you have this carboxy side chain. Okay. So, that is the fate of this reaction. But now it the reaction will, because this will not, this is not very stable again, although it is aromatic, that is why this reaction had taken place. But now you can release, it will be more stable if it is in the zwitterionic, original zwitterionic form. So, how it can do that? This H is again released, and this carbon carbon bond breaks, releasing the TPP, because that is the original starting point TPP, and in the process what you are getting is CH3 and then CO, then you have S, and this is SH, and you have this CO2H. Okay. So, this is a thioester now, thioester, but this thioester is from the reduced form of lipoic acid, because now the SS bond is no longer there. So, now there will be a trans thioesterification, a trans thioesterification. What is that? That you have coenzyme A, which is a thiol. So, this thiol now can displace the thiol that is coming from the lipoic acid. So, it is a trans thioesterification is this step, and that gives you this CH3 CO S CO A. What is released here? Released is the reduced form of lipoic acid, 1, 2, 3, 4, 5. Okay. So, this is the reduced lipoic acid. Okay. But you have got your product, what you wanted. Okay. The acetyl coenzyme A, which is the biosynthetic block for synthesis of various other large molecules like fatty acids, steroids, hormones, etc. Now, the question is, the lipoic acid which is released is in the reduced form. So, this lipoic acid molecule cannot participate in the next time, because the participation has to be by the oxidized form of lipoic acid. The participation should be by the oxidized form of the lipoic acid, and so this has to be now oxidized into this oxidized form. Say suppose this is X. So, it has to be converted into the X, because if it is not converted, then the reaction basically will need a stoichiometric amount of lipoic acid, which is not desired in an enzyme catalyzed reaction, in any catalytic reaction. Stoichiometric amount of the catalyst, where its catalyst basically is enzyme plus coenzyme. So, the coenzyme has to be also in the catalytic amount. So, there must be a regeneration process of this reduced lipoic acid into the oxidized form of lipoic acid. So, who does that? Because remember, there were three requirements of three coenzymes. One is the TPP that does the decarboxylation, then the lipoic acid that does the thioesterification, and then also this trans thioesterification by the coenzyme A, as coenzyme comes, and then that causes the trans thioesterification and releasing the acetyl coenzyme A, but in the process the lipoic acid is reduced. So, it has to be oxidized. So, there is one more coenzyme requirement, that is FAD, and FAD by the way is a coenzyme which participates in redox processes. Okay. Now, FAD is the I can again simplify the structure of FAD, no point in remembering, but you should know what are the just the important functional core of a molecule, because that is important. If you know that, then that is enough, that should be enough. So, this is, I am just writing the functional, sorry, this is the reduced form I was writing. So, this is the FAD molecule. So, there are two substituents here, and this is the R. R means that ribitol, and then followed by diphosphate, then the sugar, ribose, then the adenine. Okay. Flavin, adenine, dinucleotide. So, now this is in the, this has to be in the oxidized form, because this plus the lipoic acid SH, 1, 2, 3 SH, and then you have 1, 2, 3, 4, 5. This is the reduced form of lipoic acid. So, the product of this reaction is that FAD goes to the oxidized form, and that has got a FAD goes to the reduced form now, because this is oxidized form, I am sorry, this is the oxidized and this is the reduced form. So, when the reaction takes place, this goes to the reduced form, okay, and the hydrogens are, reduced form is addition of hydrogen. So, the hydrogens are added to both the nitrogens here and the double bond is here. So, basically this becomes FADH2. How is it happening mechanistically? So, mechanistically this is happening in this way, just one second. Mechanistically it is the, you take this one. So, again write make a box here, we start from here. So, you have NR and then double bond N, this is the aromatic ring here, and here you have again double bond N, CO, NH, CO and this. So, now you have this SH, and here you have this CO2H. Okay. 1, 2, 3, 4, maybe one more, because this is C8 system. This SH first attacks this double bond, making this nitrogen negative, which then abstracts the hydrogen, and that goes to the, so that is first part of the reaction. So, one of the nitrogen is converted to the NH. So, that job is done. So, this is N, that is R, and you have NH here and double bond N here. So, that nitrogen has not got the hydrogen yet, but in first this nitrogen is already reduced to the NH. So, now you have, what you have is AS, and then you have, sorry, CH2, then SH, and you have this carboxylic acid 1, 2, 3, 4, 5 CO2H. Now, there will be an intramolecular disulfide formation. So, this attacks the sulfur, this breaks, and this nitrogen takes up the hydrogen. So, if that happens, that means what you are getting, this is the oxidized form of lipoic acid FADH2 plus oxidized form of lipoic acid. I am just writing this way CO2H. This is the oxidized form of lipoic acid and this is a reduced form of flavin. Okay. So, this is the simple mechanism. This is basically addition of this moiety, this one of the thiol and this intramolecular attack, intramolecular nucleophilic attack forming the disulfide, resulting in reduction of the, because the disulfide means this part is oxidized. So, this part has to be reduced. So, resulting in reduction of this flavin moiety. Okay. So, that goes to FADH2. So, that is the mechanism of flavin adenine FAD mediated reaction. So, this is the mechanism by which lipoic acid is again oxidized back to the active coenzyme form. Okay. But this also gives rise to another question, that the next time when lipoic acid will be reduced, next cycle of reaction, who is going to oxidize that, because the FAD is converted to FADH2. So, now there has to be another way of making this FADH2 into FAD. So, there should be another oxidizing agent in the process where FADH2 is converted to FAD, and that is done by NAD plus, that is your vitamin B3, the coenzyme form of vitamin B3, that is this NAD, that is the nicotinic acid based dinucleotide, nicotinic acid adenine dinucleotide. Okay. We will discuss that mechanism also, or the structure will show. So, this is basically the acceptor of electrons, because it is a positively charged species. So, that is the oxidized form of the vitamin, of the coenzyme from vitamin D3, and then the reaction takes place. NAD plus brings the FADH2 back to FAD, the oxidized form, and in the process this goes to NADH. Okay. But you can again, this again will give rise to another reaction, that what happens to the NADH, because unless you have NAD plus, your FADH2 cannot be converted into FAD, and unless you have FAD, your reduced lipoic acid cannot be converted to the oxidized form of lipoic acid. So, it is a cycle of effects. So, finally, the NADH in the by the electron transfer process, it goes to the NAD plus. So, that is generated by a separate mechanism, by some electron transport processes, electron transfer processes, and there the NADH is converted to NAD plus. Okay. So, just to summarize, that what happens, that first the FAD goes to the first the TPP carries out the decarboxylation of pyruvate, then the pyruvate, then the lipoic acid comes into play and converts the pyruvate, converts the decarboxylative species into a thioester containing the lipoic acid, reduced lipoic acid. In the process, TPP is released, so that it can participate in the next cycle of reaction. Now, this thioester of lipoic acid, acetyl thioester, that undergoes trans thioesterification with coenzyme A, and then forms the acetyl CoA and releasing the reduced form of lipoic acid. Then the reduced form of lipoic acid is converted into the back to the oxidized form, the disulfide form, by FAD, and FAD in turn goes to FADH2, and this FADH2 is finally oxidized back to FAD by NAD plus, and the NAD plus, the reduced form of NAD plus, that is NADH, is then again converted into NAD plus by a separate process, which is done by some electron transfer mechanism. Okay. So, that is the the chemistry of behind the formation of acetyl coenzyme A, and this has been summarized clearly here, although it is a little difficult because the color combination is not very good. This is pyruvate. The first reaction is carbon dioxide goes, TPP comes, basically this is a TPP immediate reaction, and that holds up the acetyl group, and then the acetyl the lipoic acid has reacted whatever I have said. This is the thioester, and then this is there is a exchange that is the trans acetylase reaction, and then it goes to the again the oxidized form. It goes to the oxidized form. Who does that? FAD. So, FAD goes to the FADH2, and finally, FADH2 is oxidized by NAD plus, and then NAD plus again finally goes to the electron transport processes, and then by which it can again be regenerated into NAD plus. So, that is the full mechanism. So, it is a very complicated process, but it is a very absolute requirement for the substance for the existence of the living system. So, next in the vitamin B cycle, the B group of chemistry, B group of coenzyme chemistry, next vitamin that comes is folic acid. Okay. Now, folic acid again is a very obligatory coenzyme. It is absolutely required for the growth, for maintaining the cellular division processes. Okay. We will go back, we will discuss the chemistry, but before that let us inspect the structure of folic acid. Folic acid looks little bigger than the earlier ones, okay. Especially the pyridoxal phosphate, those type of things, or thiamine pyrophosphate. It contains a heterocyclic moiety which is called a pteridine ring. So, this is a pyrimidine fused to 1,4-diazine, okay. And then pteridine ring is attached by a CH2 to what is called para-aminobenzoic acid. This is what is para-aminobenzoic acid. And then this para-aminobenzoic acid, the acid group is attached by an amide bond into the glutamic acid moiety. So, this is basically a pteroyl para-aminobenzoyl glutamate or glutamic acid, that is what is folic acid. Okay. Now, the numbering system goes like this: this is 1, this is 2, this is 3, this is 4, this is 5, this is 6, 7 and 8. Okay. So, now interesting fact about folic acid is that, now this is not the coenzyme form, this is the vitamin. Okay. So, this is the vitamin, so this has to be transformed into the actual coenzyme form. Now, bacteria can make their own folic acid. Actually, bacteria always makes their own folic acid. In fact, it cannot take up any folic acid from outside. See, if there is exogenous supply of folic acid, it will not utilize that. It will always make in its own cell. It has got a biosynthetic machinery to make the folic acid. On the other hand, we humans, we are part of the mammal. So, these humans, we cannot synthesize folic acid, but this folic acid has to be taken from outside, that means through diet. So, through our diet we take the folic acid. Okay. So, there is a difference between the bacteria and the human. Next is that folic acid, what is the active form of this coenzyme form of folic acid? Let me see whether it is shown here somewhere. The active form of folic acid is not shown here. I will write that. The active form is, see, folic acid is, you should practice writing the structure, but do not memorize it in that way, but always try to find out what is the important functional moieties in the in folic acid, or in fact in any molecule which has to be there in your mind, otherwise it will be very difficult to progress. Okay. Now, but folic acid, the problem is all these all these are quite functional or act as functional parts in folic acid. So, you have to remember, unfortunately, it is better that if you remember the structure of folic acid. Now, what is the active form of folic acid? It is this one, that this is NH, this is NH, and then you have CH2, this is NH, this is your para-amino benzoic acid, and that is attached to glutamic acid. So, that means it is, again I write 1, 2, 3, 4, 5, 6, 7, 8. So, this is 5, 6, 7, 8. 5, 6, 7, 8 tetrahydrofolic acid, I can write THF. This is the active form of, or active means the coenzyme form of folic acid. What type of reaction it does? It participates in reactions, means it acts as a coenzyme supporting the or helping the actual enzyme to catalyze a reaction in which a carbon, one carbon is transferred from a substrate into another substrate and resulting in the product. Okay. So, basically it participates in one carbon transfer, one carbon transfer. Okay. So, one carbon transfer reactions are very important, very important. I can give you an example that in RNA you have uracil and in DNA you have thymine. Okay. Now, uracil, difference between thymine and uracil is basically one carbon, one methyl, and in fact thymine is derived from uracil uracil monophosphate, deoxyuracil monophosphate, uridine, that is called uridine monophosphate, UMP, uridine monophosphate. That means you have the uracil, then the deoxy sugar, and then you have the phosphate. So, that uracil is converted into thymine deoxyribosyl phosphate. That means that is called DTMP. So, DUMP deoxyuridine monophosphate, that means here you have this OH, you have this phosphate, and here you have uracil, that is converted into the thymine T, okay, phosphate, and here the OH. So, that is the reaction. This is a very vital reaction, because unless this thymine TMP is this is DTMP, unless this DTMP is synthesized, you cannot get the building blocks of your DNA. Okay. So, that is extremely important and very interesting, because that can give rise to what are called anti-cancer compounds, because if you can stop this process, then you stop the biosynthesis of DNA. If DNA cannot be made, that means the replication cannot be done, and if replication cannot be done, ultimately cell cannot divide and ultimately it has to die. So, no growth of the cell. Okay. So, that is what happens here. So, now just have a quick look at the how bacteria makes its, because we are not making the folic acid, we get the folic acid from diet. So, that chemistry is little bit easier. That folic acid has to be converted into the active coenzyme form. In the bacteria microorganisms, it is GTP, that means the guanosine triphosphate, GTP. Okay. The GTP is by a rearrangement process, degradative rearrangement process, this is a little bit complicated. So, we are not going into that, is converted into this pteridine nucleus, but this is, remember, this is a dihydropteridine, this is the 7-8. So, first product in the biosynthetic pathway is this pteridine nucleus, but that is containing, this is a dihydropteridine nucleus, that is the first one. The second is this OH has to be replaced by para-amino benzoic acid, but OH is a bad living group. So, that has been converted to a phosphate or pyrophosphate. In fact, it is a kinase enzyme which does pyrophosphorylation. The pyrophosphorylation makes it a very good living group. Now, in presence of another enzyme, the para-amino benzoic acid, the NH2 comes and attacks here, kicking out the pyrophosphate, and this is the para-amino benzoic acid now is attached to the pteridine nucleus. Okay. So, this is not yet the folic acid, yet you did not get. But remember, you started all the time with the dihydro, 7,8 dihydro, and then the third enzyme in this biosynthetic machinery. We are not talking about this process. There are enzymatic enzymes involved here. Conversion of guanosine, this is ACE, guanosine triphosphate, that is conversion. That conversion we are not discussing. So, this is one enzyme, that is another enzyme. First enzyme converts this to pyrophosphate, second enzyme displaces the pyrophosphate by para-aminobenzoic acid, and the third enzyme you get the what is called, this is not tetrahydrofolic acid, this is the dihydro 7,8 dihydrofolic acid. Dihydrofolic acid. Okay. Now, this 7,8 dihydrofolic acid, now because your active coenzyme form is this one, 7,8 dihydrofolate has to be reduced, this double bond has to be reduced in order to get to this fully this saturated form here. So, that is by an enzyme, a fourth enzyme in the machinery, that is called dihydrofolate reductase, and dihydrofolate reductase ultimately gives the coenzyme form of folic acid. Okay. So, these are the four important enzymes. One is the kinase, another is this the PABA the incorporation of PABA, and then the third one is this incorporation of glutamic acid, and the fourth one is dihydrofolate reductase, by which you will get 5, 6, 7, 8 tetrahydrofolate. What about in humans? The humans, from diet you get folic acid, that means double bonds here as well as here. So, first there is an enzyme called folate reductase. This folate reductase forms the dihydrofolate, 7,8 dihydrofolate. So, folate reductase can reduce the double bond in folic acid, 7,8 double bond in folic acid, to form the 7,8 dihydrofolate, and the 7,8 dihydrofolate under the influence of the dihydrofolate reductase DHFR, but this DHFR belongs to the human. So, this is a human dihydrofolate reductase that converts the 7,8 dihydrofolate into the tetrahydrofolate, 5, 6, 7, 8 tetrahydrofolate. Okay. Remember, this when it occurs in bacteria, this is the bacterial dihydrofolate reductase. When it occurs in the human, it is the human dihydrofolate reductase, and when it occurs in a parasite, so that will be a parasitic dihydrofolate reductase. Why I am spending so much time on these biosynthesis? Because this folic acid biosynthesis, understanding the folic acid biosynthesis has given rise to different types of drugs. One is antibacterial drug, because if you can stop producing this dihydrofolate from the in the bacteria, you will get antibacterial compound. If you can stop if the dihydrofolate reductase, although the enzymes do the same reaction, but there is some difference between the bacterial dihydrofolate reductase versus human dihydrofolate reductase versus the parasitic dihydrofolate reductase. So, by selectively inhibiting one of these, you can get anti-parasitic compound like anti-malarial compounds, and if you can selectively inhibit the folate this dihydrofolate reductase in human, you will get an anti-cancer compound, and if you can selectively block the dihydrofolate reductase in bacteria, you again get antibacterial compounds. Okay. So, there are 3 different classes of drugs that you can get. Some names are given here: pyrimethamine, that is an anti-malarial agent, then sulfonamide, which is a first antibacterial agent to be used, rationally designed before this is after Paul Ehrlich's discovery of arsenic based arsenic based compounds, antibacterial compounds like salvarsan, and this is another drug called trimethoprim. This is trimethoprim, that is an antibacterial compound. Okay. So, and there is one more compound, anti-cancer compound, that is called methotrexate, and you will see methotrexate has a structure which is very similar to the folic acid. Folic acid has a carbonyl here, here it is the NH2, and folic acid has here NH, and instead of NH there is a methyl here, but it's a it's a structural mimic of your folic acid, right. So, but this acts, it is a very famous, in fact the first anti-cancer compound developed, anti-cancer compound. Okay. So, next session, the next session we will discuss the how these type of compounds have evolved, and then finish up the coenzyme chemistry by so what is remaining is the vitamin K chemistry plus some other B group of coenzymes like cyanocobalamin, that is the B12. So, some chemistries are left, biotin, that chemistry is left, and but we will do that in the next session. Thank you.